I'll introduce the paper submitted to iMetaOmics. The title is Lactobacillus johnsonii-FM1 modulates gut microbiota and secretes anticancer metabolite vanillic acid to inhibit colorectal tumorigenesis. Probiotics can mitigate the progression of colorectal tumors by maintaining gut microbiota homeostasis, restoring the integrity of the damaged mucosal barrier, promoting cancer cell apoptosis, enhancing the host immune response, and counteracting oxidative stress. The highlights of this article are mainly as follows. Now I will present the results. To investigate the effect of L-johnsonii-FM1 on colorectal tumorigenesis, APC-Min plus mice were orally gavaged with L-johnsonii-FM1 E. coli or PBS. Mice receiving L-johnsonii-FM1 showed significantly fewer and smaller tumors, with markedly reduced cancer cell proliferation. These findings highlight the anti-tumor effect of L-johnsonii-FM1, forming the core of our results. Probiotics can enhance microbiota diversity and restore gut balance, helping to slow colorectal cancer progression. To test this, we performed metagenomic sequencing on APC-Min plus mice treated with L-johnsonii-FM1 E. coli or PBS. L-johnsonii-FM1 increased beneficial microbes, reduced harmful ones, and preserved microbial homeostasis. Next, we further investigated the effect of Lactobacillus johnsonii-FM1 on gut barrier function in colorectal cancer model mice. The results showed that L-johnsonii-FM1 significantly alleviated intestinal barrier dysfunction and maintained barrier integrity by suppressing inflammation levels. To test its anti-cancer effects in vitro, we treated CRC cells and normal colon cells with L-johnsonii-FM1 supernatant, using E. coli supernatant and brain-heart infusion as controls. Compared to both controls, L-johnsonii-FM1 significantly reduced cancer cell viability and colony formation, without harming normal cells. Given the importance of microbiota-derived metabolites in health and disease, we aim to identify anti-CRC compounds produced by L-johnsonii-FM first. Untargeted LC-MS of fecal samples from treated APC-Min plus mice revealed vanillic acid as a key metabolite. This finding was confirmed by targeted analysis of the L-johnsonii-FM1 culture supernatant. We evaluated the functional effects of VCA in CRC cell growth in vitro and in CRC tumorigenesis in vivo. In vitro experiments showed that vanillic acid significantly inhibited the proliferation of colorectal cancer cells and promoted their apoptosis. In vivo studies further demonstrated that mice treated with vanillic acid exhibited a marked reduction in both the number and size of intestinal tumors. To further investigate the potential mechanisms by which VCA antagonized colorectal tumor progression, we conducted RNA sequencing on the colon tissues of VCA-treated mice and control mice. The results showed that the expression levels of known target genes or downstream effectors of the WNT β-catenin signaling pathway were significantly reduced. To test if VCA works through WNT β-catenin, we used CHIR 99021 in HCT116 cells. VCA blocked CHIR-induced activation, reduced proliferation, and promoted apoptosis, confirming WNT beta-catenin involvement. In summary, Lactobacillus johnsonii-FM1 restores microbial diversity, strengthens the intestinal barrier, and reduces inflammation. It produces vanillic acid, a key metabolite that triggers tumor cell apoptosis and blocks WNT beta-catenin signaling, ultimately suppressing colorectal cancer progression.